Welcome to the lecture on MHC Class 1 Antigen Processing and Presentation. For today's session, we have 4 learning outcomes. First, list the steps in antigen processing and peptide generation. Second, recall how peptides are transported into the endoplasmic reticulum. Third, explain the assembly of the MHC1 loading complex. And lastly, describe how MHC1 antigen complexes are presented on the cellular surface. Let's look at this process in greater detail. For this, we have our model cell shown with extracellular and intracellular compartment and some of the organelles that are participating in this process. So we already have our heavy chain that is membrane residing expressed, and also our beta-2 microglobulin, which is soluble. All right, and so we have a cell that then is infected with either a bacteria or a virus. So bacteria could be Salmonella or also tuberculosis. Those can reside intracellularly, and this generates a lot of foreign particles that are degraded and ubiquitinated. And so the step of these peptide generation and trimming is mediated by the proteasome, which has enzymatic active beta core. So this is a heptamer ring. And there are different activities of proteases that then will chop down the polypeptides. And interestingly, when we have an infection and inflammation ongoing, there is also interferon signaling occurring, and this will then change the subunit composition of proteasomes, which will be expressed in response, and that then will turn into an immunoproteasome. This immunoproteasome has a way higher activity processing these peptides. Those polypeptides are then further processed and broken down in order for them to be able to enter the endoplasmic reticulum. For the next step, our peptides will have to be transported into the ER lumen in the proximity of the MHC1 complex. And so for that, we have a specific transporter, TAB1 and TAB2, which is an ATP binding cassette transporter. And this transporter favors 8 to 12 residues of peptides that can be transported in. Larger ones will be further broken down by peptidase in the cytoplasm, but also in the ER is a specific one that facilitates that. Next step involves the assembly of the MHC1 class loading complex, and there are several chaperones that are involved in this peptide loading process, and those help to stabilize it. So calnexin, for example, also membrane residing, is one chaperone. Calreticulin, both are calcium dependent. And then there is also ERP57, which is the oxidoreductase. And so calnexin interacts with the heavy chain, and it allows this to be in concert with the oxidoreductase to have the right oxidation state and then be conformationally favorable in order to bind the beta-2 microglobuli. Calnexin then dissociates and gives the MHC1 heterodimer an open form that interacts with calreticulin. Importantly, peptide binding then requires the additional participation of TAP as well as tapasin in order to assure a pool of free peptides which are in proximity. This peptide loading complex stabilizes then the MHC1 molecule, so that's receptive here to peptide binding and mediates the exchange of low affinity to high affinity binding. The process that ERP57 and tapasin mediate is called peptide editing here, and ERP57 may do this by breaking and reform disulfide bonds in the alpha-2 domain during peptide loading. Antigen binding of the peptide, the chaperones then are released, and the peptide MHC1 complex is moved across the ER within vesicles, and then further across the Golgi, where it's then fused to the plasma membrane and presented at the plasma membrane. A CD8 positive cell then can interact with the T cell receptor and initiate the cascade of apoptosis. Thank you very much. Here is some of the literature cited, as well as further reading, in particular if you're interested in how defects in those pathways contribute to immunodeficiencies, but also to the development of cancers. Thank you.